There's a lot of discussion among biologists that there's this idea that if an animal is charismatic, if it's a polar bear, or if it's a fluffy, cute thing, then there's a lot more incentive in the community, in the public, to conserve that organism. So sometimes it's harder to back up funding for conservation of organisms that, are not necessarily soft and fluffy. So I'm going to tell you about some of that work today. So the title of my talk, as you can read yourselves, is Sight Unseen, Using Environmental DNA to Detect Native Freshwater Pearly Mussels. So I'm going to start off my talk by telling you a little bit about my organism and genetics, and then talking to you about what my findings were. So the aims of my project were to develop genetic tools, so I'm going to use the word markers a few times, I'm going to explain that in a second, to detect 4 native mussel species at risk from environmental water samples. So eDNA markers are just a part of the genome of an organism, so normally a small section of a gene that's unique in some way, so it can be used to identify the species, or it can be a way to track down what's going on in their genetics. So a marker is just a section of the DNA. So environmental DNA is DNA that's been extracted from water samples in this case, or it could also be soil samples, that were collected at sites with presence or absence of the organism. For my study, because I wanted to develop these tools for the first time, and then to test whether or not they were working, I collected all my samples at river sites where we knew the mussels already existed. So it's considered sight unseen because you do not actually have to interact with the mussels at all in order to do this sampling. You simply take a water sample, and then you extract their DNA from that sample. So the DNA gets out into the environment from a couple of different sources. One is just degradation of gametes and larvae. So when males broadcast spawn, they're just releasing sperm into the water column, and that will degrade if it's not actually picked up by a female, and that will release DNA. Also just living their daily lives, mussels are filtering water, and as they're doing so, they're releasing cells into the water. And then finally, if an organism dies, as it degrades and it decomposes, you'll also have DNA being released. So I'm going to talk to you guys a little bit about this plot, but do not be overwhelmed. So I designed a different marker system for each of my 4 species. So each one is targeted. So if you use the one and it detects something, you know that that links up to the presence of DNA from that particular species. So my readouts actually look like this. This is a set of standards. So these are, can you see them as? No, you can't. Okay. So each of these bands represents 1 concentration of DNA. So that's just so that I can then reference back the samples that I had and know how much DNA was in those samples. So many of you have probably heard about PCR. It's just the amplification of DNA, making new copies. So every cycle of PCR, you get doubling of the DNA. In this case, there's also fluorescence involved. So every cycle of PCR, you get a little bit of light being released by the reaction. And so the more light there is, the more DNA is in your sample. And so if you have a curve like this forming, that means that your tool is successfully detecting DNA from your target species that you're looking for. So my study species, I had 4 species, and they were all in the family Unionidae. So these are the freshwater pearly mussels. These don't include things like zebra mussels and fingernail clams and the invasives. I only looked at natives. They are filter feeders, so a lot of people think that they consume detritus, things that are just decomposing in the water. That's actually not true. So they're very selective about what they eat, and they prefer bacteria and algae. And many of the species in Ontario are either threatened or endangered. About 1/3 of species in Ontario are endangered. Can you guys guess about how many species of Unionid there are in Ontario? Native mussels. I know a couple people know the answer already. Do you want to throw out a guess?
42.
42? Oh, you're so close. Yeah? 80. So, if you go out most people when they go into a river, they notice zebra mussels, right? Because they're hugely prevalent and they're in really big numbers. But most people don't know that we've got a really wide range of native species. So the exact number is debatable. People estimate from about 40 to 42. So that's because there's one, yeah, that was a great guess. So one of the species was found as a degraded shell that was in really bad condition, and it's never been found again. So most people don't count that one. So most people say 41. They are also, because they have been impacted so severely by a couple of impacts I'll tell you about later, their distributions are quite small. So a lot of the species that are existing in Ontario that are endangered only are found in Ontario in the northern United States, or only in Ontario and Manitoba. They also have obligate parasitic larvae. So what this means is after the eggs are fertilized by the sperm, the females brood them for a little while, and they form into these little clam-like structures that are very, very cute and can only really be seen clearly under a microscope. And those larvae are then, they then infect, it's not really the word I want to use, but they do infect a host species. So it's usually a fish, but it can also be a mud puppy mussel. A mud puppy. There are mud puppy mussels. There's one species that infects a salamander. And because of this really important behavior where they need to attach to the host in order to develop and also to disperse, they have some really cool host attraction behaviors. So these are two of the behaviors, but there are actually three that you find in Ontario. So this first one, you see two fish right here, and then you see a third fish. That fish is not a fish. That is actually a part of the mussel. So when the female is ready to release her glycidia, when they're fully mature, a part of her inner flesh opens out, and it looks like a fish, and she even waves it in the water so it looks like it's swimming. And this attracts larger predatory fish who go and attack, and when they attack, they get a really unpleasant mouthful of glycidia. So that's one way. And then another one here on the right, this is actually just a pouch of glycidia. So it's made to look like an insect larva, but in fact it's not a larva at all. Those are fake eye spots, and it's just full of glycidia. And then the third way you see in Ontario is there's a mussel that actually clamps down on the nose of a fish and slowly releases the glycidia into its gills. So that's the snuff box. So, Unionidae have a lot of different values for our ecosystems. Of course, we always hear about biodiversity, and biodiversity is really important. Ecosystems are a lot more resilient to pollutions and other degradation when you have a lot of diversity. But they're also part of our food chain. So there are several different native mammal species that eat them. And historically, they were also used as mother of pearl. So that is one of the reasons why they are at such low numbers and why it is hard to find them, is that they're highly over-harvested historically. And then finally, they're ecosystem engineers. So what this means is they have a really important role in shaping the way that the aquatic ecosystem looks. And they do this by improving water quality and clarity. So they can filter approximately 40 liters of water in a day. So they're actually doing a lot of work to improve the aquatic ecosystem in their areas. And so although I told you that historically overharvesting was really influential for their declines, at the present moment it's actually zebra mussels that are having the most impact. Zebra mussels will actually bind onto the native mussel and it'll hold them shut so they can't feed, and they'll just starve themselves to death. So it's rather sad, but that's another reason why it's important to distinguish between our native mussels and the invasives. And that's also why it can be difficult to eradicate one group, because we do have native species that are fairly closely related. So I looked at 4 target species, but there's no reason why the same technology could not be expanded and applied to other species. I just targeted these ones because they are all species at risk. So the rationale for my study, mussels are imperiled, as I've told you, so they're either endangered or threatened, and there's a lot that we still don't know about them. And one of the reasons for that is that they're very hard to find and identify. So this is a picture of me standing in one of my sites, and this is one of the best sites in Ontario. It's got tons of mussels, it's great. As you can tell, the water clarity is not ideal, and this is actually at a point where there was really low flow. When I went there earlier in the year, the banks were completely overflowing, and the water would have been above my head. So it would be impossible to go and sample mussels in those conditions. And then also in this picture, just off the top of my head, I counted 5 species. They all kind of look like dirty rocks. So when they're in the water, they come out and they're very much covered in algae and a lot of debris. So they are hard to distinguish if you don't have the expertise to do so. So traditional sampling does involve going in, pulling them out, and trying to ID them. And so this is really effective. You can figure out whether the species is there, but it's time consuming because you need a crew of people out there, especially for water safety. And it's also expensive because in order to sample a single site using quadrat sampling, where they dig up 1 meter by 1 meter squares, it'll take a crew of about 3 or 4 people at least 2 days. So it's just expensive to pay those people for that much time. And then finally, there's a disturbance of the habitat. Every time you're going in, you're digging up your mussels. So when you're working with species at risk, ideally you're not disturbing them. So I had a few study questions, but I'm only going to talk to you about 2 of them today. So one was, can targeted eDNA tools be developed? So the reason why this was a big question up in the air at the time, when I started my project, was that we're trying to design species-specific primers, so ones that only amplify one species among 41 species, right? So these are all close relatives to each other, which means their DNA looks really similar. So the closer their DNA is to each other, the harder it is to distinguish them. And then I wanted to know whether eDNA was able to detect species at a similar frequency to our traditional methods. So my study sites were predominantly in the southwestern Ontario area, across 6 different rivers, but there were a couple of sites to the east, and I selected those because one of my species is only found in the east. So this graph, along the bottom, these are all the sites that I sampled, all the ones that were in the west, rather. And then the height of the bar indicates basically the success of the tool. So what proportion of the samples that I got back at a site allowed me to identify my species. So the blue bars are quadrats. So this means that about 30% of the quadrats that were sampled, you actually pulled out an individual of that species. And then eDNA means that of the qPCR cycles that I did, this is the proportion that came back positive for my species. So I'm going to go a little bit faster because I think that was my time's up. No, oh, it wasn't? Okay, good. So good, because I've got so much more to tell you. So a couple of neat things here. One is that at every site where there's a blue bar, every site where we found the mussel using traditional sampling, we also found the species using eDNA, which was huge, because at the time that I was doing my research, we weren't even aware whether mussels shed enough DNA into the environment to be detected. Another thing that you should notice is that some of these are blank. So this means that all the sites where we did not expect the species to be, where it did not make sense for them to be, they were never detected, which means that I'm not detecting some other species that look similar. I'm probably actually detecting the species I'm looking for. And then finally, these orange bars are quite tall. So this is showing that a high proportion of my samples are coming back positive, even when there are very few mussels being found in that river. So it's very sensitive. And in fact, I was able to detect my species at 2 new sites where it had not been found by traditional sampling methods. And these are both supported by previous beliefs over what had been there, or other sampling that people had done by the shore finding remnants of the species. So this is another neat thing that was actually an accident that came about, a happy accident. I told you that when I went to the sites earlier in the year, it was really dangerous to sample, and that's because we had had a couple of really huge storms. So I actually ended up having to cancel my sampling and go back again later in the year. But while I went in that first week, I was able to collect samples at a couple of sites, and luckily nobody fell in the water and drowned before we canceled it. And so one of my sites I sampled twice. And so what we can see here is in the regular season, where I showed you a picture of myself in the water and it was weightable, it was still high but weightable, I was detecting my species in 83 to 100% of my samples, which is huge, right? That means that I'm getting really consistent detection. But even when the water flows were so high that I could barely get into the water, I was still getting detection in at least 2 out of my 12 replicates. So this is a really good sign. And then it becomes more impressive when you realize that across the 75 square meters that were sampled at that site, only 8 individuals of that species were found. So that species goes from about this big up to about that big. So 8 of these mussels across a huge river, and I'm still getting detection in multiple samples. So that really speaks to how sensitive this tool is. So eDNA, I don't think will ever replace traditional sampling. I think it'll always just be a tool and a way to target where we want to do the more intensive sampling. But it's still going to be an effective tool for doing that. So a couple of things that we can't get from eDNA sampling are sex ratios. So you can tell that an organism is there shedding DNA, but you don't know whether it's female or male. And there are 2 species at risk in Ontario that we already know have too many males. So if there are a lot of males of a species and very few females, they're going to have really low reproduction rates because only so many glaucidia can be developed by each female. You also don't know whether or not the individuals that are there are too old to still be reproducing, or if they're too young and they're not reproducing yet. You also can't tell whether there's still young being introduced into the population. So So if you get a lot of the smaller size classes, you're finding really small mussels, then that means that the younger mussels are still being brought into the population and the population is still growing. Whereas sometimes you find something that's called a functionally extirpated population. So this means that they're still there, you can still detect them with eDNA, but they're not actually reproducing anymore. So there's also some belief that the more eDNA there is, the bigger the population is, but it's not a direct correlation. So that's another thing that you can't tell. But what you can do is you can detect these mussels without disturbing their habitat. You can collect water samples from downstream of where you know that they are, or you can collect water samples from a boat so you're not actually traipsing in amongst their habitat. You can then go out and sample a very large area because it's a very cheap method compared to the intensive sampling with a crew. And it's also so sensitive that we were able to detect the species, one of the species at 2 new sites. So it means that it might even be more sensitive than traditional methods. And so this will allow us to sample very rare species or very small populations. And finally, I'm excited about this idea that you could get more integration into the community and get community members to act as citizen scientists, and so they could go out and collect samples for researchers and then send those samples to be analyzed. So that allows us to collect samples from a much broader range of areas without having to pay researchers to go driving. This is a quick summary that is not particularly important, but basically, eDNA, there are a lot of issues where people are concerned about contamination. They say, okay, you said that you found it, but what if it's just contaminated DNA from somewhere else? Was that species really there? All of my controls, which are all negative samples that I collected, hoping that there would be nothing in them, almost all of them were actually negative. So that means there isn't a lot of contamination. And I was able to develop successful eDNA markers for 3 of my 4 species, and the 4th one I think I'm going to figure out pretty quickly, but there were some issues with it that still need to be worked out. Sampling from the surface of the water was equally as effective as sampling lower in the water column. This isn't something I told you about with my methods and my goals, but this is something that I addressed as just a piece of interest because some researchers have shown that benthic organisms that live at the bottom of the water have more DNA at the bottom of the river, which makes a lot of sense. I didn't actually find that in my study. And then, like I said, eDNA copy numbers are correlated with population size. If you've got a huge population, you do see more eDNA, but the correlation was pretty weak because it also depends on how much water is flowing by and that sort of thing. And then, like I told you, under the really high flow conditions, we were still able to detect species. So that's really interesting. Lots of people helped me in my research, so thank you to all of them, and thank you to all of you for being here today.
So you just touched on this about being able to detect the mussels downstream from where they actually occur, and I was wondering how you tested that, how far away might you be detecting them, and if they occur in some other tributary, would you detect them downstream?
So I didn't test that, and that's one of the things in my thesis around fusion interactions. This would be really interesting to know. So based on my research, it doesn't look like there's a huge range of detectable flow range. So although the DNA, of course, will flow down into the lake, it dilutes enough that you don't actually detect it very far downstream. And that's based on the fact that one of my species wasn't found at any of my sites that I sampled, but I know that upstream of there is a population, and I didn't detect that population. So it's anecdotal, but it would be really interesting to have somebody actually study that. Yeah. Sarah first.
Just a method thing. What do you have to do for your negative controls? Is it just water, or what do you do?
Okay, so negative controls. The first negative control you have is tap water in a jar that you put in with all of your samples before you go out sampling for the day. And that's called a cooler control, and it's the idea that your cooler itself might be contaminated. And then you get back to the lab and you have filtration controls. So you're just pouring, again, tap water into all of your filtration apparatus, and then you run your samples, and then you do the same thing again. And then you have, every time you extract your DNA, so those little filters that you filter all the water through, you then have to get the DNA isolated from that. So that process, again, it's an extraction negative, so that's lysis buffer. And then after that, when you're doing your cocktail, you've got negatives in there. So there are a lot of negatives, and the reason for that is that every time you get an unusual result, you want to be really certain that you can back that up with a negative and say, okay, this unusual result is not caused by contamination. If I didn't have as many controls and I had found that my species was in those two insoluble river sites, I would have been like, well, maybe it's contamination, who knows? But because I have all those controls, I can be more confident. Does that answer your question?
Yeah, just because it was brought up in your defense, you had a lot of controls.
A lot of controls. Yeah. Oh, and then Michael, I think next.
Yeah, well, that was great. Really enjoyed that. See why that was dependent so well. My question is more the arty guy question, and I don't know much about controls or mussels in general. But you said they're charismatic, but you were across the whole province from the south to looking for these guys. Did you find migration patterns? Did you find surprises that they were more charismatic? Perhaps they got around more. Maybe they did the 6, or how many steps there are there to sexual reproduction? Maybe they were doing the steps and they were finding their way, up to Eastern Ontario and around.
I'm sure males and females of the same sex find each other very charismatic. So that's not the issue, right? It's that, how many of you already knew that they had mantle displays and that they create these little things that have eye spots and are so convincing as larvae, right? There are all these really neat things about them, and there's something I didn't tell you guys about, which is in a normal cell, like all of us, we all have our maternal mitochondria. So this means that we've got little organelles in all of our cells, and they contain DNA, and all of us have our mothers. So none of us got anything from our father because he would have only inherited his own mother's mitochondria. In mussels, this is the only group that you see where there are 2 types of mitochondria. So this is something where, if we didn't study mussels, we would have never found this super unique genetic inheritance system. And so, that in itself is incredibly interesting. If you look at the mitochondria of a male and a female of the same species, they're less closely related than a male and a male of different species. So the mitochondria diverged long before these species became species. So this kind of stuff is so neat, right? But to answer the second half of your question, there isn't a lot of dispersal in them, and the reason for that is that the only point where they can disperse is when they're attached to their host fish, right? So if you have fish that are only traveling short distances within rivers, they're not going to be able to go into Lake Ontario, move over, and go up another river, right?
So they're very charismatic, but they just don't get around that much.
Poor mussels, they don't get around. But I think that the more we talk about some of the amazing wildlife that we have in Ontario and that we have in Canada and the world in general, I think that we can get citizens really excited about it. It's just that people don't know, right? I didn't know how cool mussels were until I started studying them. I think you had this.
Just a methodological, kind of base question, but how much water is it that you take in that you run the analysis on?
So it's actually a shockingly small amount. So at every site I sampled 12 1-liter bottles. All of those bottles were filtered through a filtration apparatus, and then each of those filters became 100 microliters of extract. So when you do all the calculations, you go back to what it would have been. Each of my detections was from about 25 milliliters of water. So that's 1/10 of a cup. And I was still getting detection in 2 out of 12 of them with 8 individuals in the whole river, or in that whole section of the river. So, and then I think there's one back there.
Will we ever get to the point where we've refined or we've advanced the DNA amplification system enough that we can identify chromosomal sex chromosomes, the chromosomal differences, to be able to identify males and females in a population?
So the reason why that's an interesting question is there aren't sex chromosomes in mussels. So all you have is the male mitochondrion and the female mitochondrion. So both males and females inherit both from their parents, and then by differential expression from the maternal line and all these different things go on, and basically you lose one or the other. So when you lose one or the other, whichever one's left over, that's the sex that the mussel becomes. So it is possible to go out and sample eDNA and develop markers that recognize the male sequence and markers that recognize the female sequence. But all that would really tell us is, are males here and are females here? Because you can't then back calculate and say how many mussels are there here. That's something that you really need to go to the site and sample using traditional methods.
My question is less specific to your research in particular and more just kind of biology in general. And I was curious because you mentioned invasive species earlier and how the mussels will clamp onto the native species and kind of starve them to death. And then you also mentioned biodiversity and the benefits of that. So I guess my question to you is, assuming the biodiversity of a site remains at the same level, even with the influence of an invasive species, how problematic is that invasive species really if the biodiversity is staying the same?
So some of that comes down to nomenclature. So generally an invasive species will only be considered invasive if it's having a negative impact. Okay. So there's a difference between introduced species and invasive species. So in the case of zebra mussels, we can now see after a site has had zebra mussels for about a decade, you almost can't find native mussels anymore because it's killed off all of them. So like that's where we say, okay, that's a huge impact. We know that it influences plant life in rivers. We know that it has all these negative influences, so that's why it's considered invasive. That being said, there is this philosophical debate as to is it xenophobia? Is it us saying, oh, it's introduced so it's less worthwhile? It's still a species that is living in this area. Doesn't it deserve to be protected? And so I guess the one counter argument to that is most species are introduced for quite a while before they become invasive. So usually you'll be able to detect a species for several years or several decades before we start to see, oh, wait a second, they're having huge impacts now, because it really takes time for them to get established. So that's where it might be better to get rid of an introduced species before it's so established that it starts having the negative impacts. So if that answers your question.
No, that answers it perfectly.
Any other questions? Yeah.
How do you feel when you see people eat mussels?
So most people do not eat the mussels that I study. So apparently marine mussels taste far better than
We eat saltwater mussels.
Yes, everyone eats saltwater mussels.
You don't care about those.
And it's actually funny because I did my defense yesterday, and the chair of the session, this question came up, and he said, "Oh, I've eaten freshwater mussels. They were, well, different, but I ate them." So from that, I'll take it as they're not particularly tasty. But I mean, I don't eat animals, so like I've got my own biases on that. I think that as long as there's sustainable harvest, I don't see an issue with eating animals. But obviously, if you're trying to eat our native mussels that are already declining, that I would have an ethical dilemma with. Yeah.
For the sites that you went to, they were mostly rivers, or did you look at lakes as well?
I only looked at rivers.
Okay. For this process, the DNA, does it work better in kind of flowing rivers where there's a lot of that like circulation, or would it work just as well in kind of a lake where it's a bit more flat?
That's an interesting question. So my mussels, it wouldn't have worked very well to sample lakes because you only really see them in wetland areas of lakes. So you don't, generally the rule is that they're not in lakes. But you will sometimes find them along edges. So there's that. There's definitely this idea that in a lake or in a pond, you're going to have the DNA settling down through. So if you're sampling off the water, you might get less of a detection because the DNA is just settled. But then there's also the idea that in a pond, your DNA, it might be settling, but it's constantly building up, and at a certain point, it's going to saturate the lake, right? There's going to be a lot of it. Whereas with a river system, every bit of DNA that's shed gets washed down the river and ends up in the lake, right? So you're constantly losing that DNA. So better or worse, I think it's always going to depend on how fast your flow rates, how much DNA is being shed into the environment, what type of organism is it. Might plants be more difficult to detect than animals? Like, there are all those other questions. So I don't think I've ever seen a direct comparison where someone said detection is better in a river than it is in a lake. I think they're just different systems and you're going to have to work with them differently.
I guess for the most part, species that live in rivers and species that live in lakes are usually a bit more distinct. So. Sometimes
fish, you have some overlap. And so I've seen people sample the same organism in the lake and the river. But I don't think they made the comparison and said, it could be that I just didn't, get that far in the literature because I was looking at rivers.
Okay, that's perfect. Thanks, Charise.